Hi students, this is A. Mandri Kumar Rajesh. In this video, we are going to discuss a topic called screening for recombinants. That means after the introduction of recombinant DNA molecule into the suitable host cell, we have to identify those cells which receives the recombinant DNA molecule. So we have to do a process called screening. If a cell which receives the recombinant DNA molecule, they are able to express the trait or character. Suppose if the cell which do not possess or receive the recombinant DNA or the foreign DNA, they are not able to produce that character, or they are not able to express that character or the trait. So in our book, they have given three screening methods. They are insertional inactivation method or blue-white colony screening method. Then the second one is antibiotic resistant markers. Then the third one is replica plating technique. So the three methods are insertional inactivation method or blue-white colony screening method. Then the second one is antibiotic resistant marker. The third one is replica plating technique. Let me teach you these three methods one by one. First, I am going to tell about the insertional inactivation method. This insertional inactivation method is the most efficient and rapid method to identify the recombinant DNA or the recombinant bacteria. Then this method mainly relies on the activity of the enzyme beta-galactosidase. Occurs in the bacteria E. coli, which can cleave the lactose into glucose and galactose. So this method mainly relies on the activity of the enzyme beta-galactosidase. This enzyme which is present in the bacteria E. coli, which can cleave lactose into glucose and galactose. So the beta-galactosidase enzyme which can cleave the lactose into glucose and galactose. So the presence of lactose in the environment which triggers the bacteria E. coli, which triggers the lactate operon, in the bacteria E. coli. Then these operons are able to produce the enzyme beta-galactosidase. Then this beta-galactosidase enzyme can metabolize lactose. So most of the plasmid vector carries the short gene segment of lactate gene. So most of the plasmid vectors carries the short segment of lactate gene, which can able to produce that amino acid beta-galactosidase, which are able to produce the amino acid of beta-galactosidase. Then here you can find a plasmid vector which contains antibiotic resistant marker, that is the selectable marker, and which contains multiple cloning site. Then the multiple cloning sites are present within the lag sequence. So that multiple cloning sites are present within the lag Z sequence. That means this multiple cloning sites may cleave by the restriction endonuclease. Then a foreign DNA are able to insert in this cloning site. At that time, that insertion make the lag sequence, the lactate sequence inactivate. So there is no appearance of blue color because the enzyme beta-galactosidase are not produced by the lactate sequence, because that foreign DNA inactivate this lactate sequence. So they are not able to produce the beta galactosidase enzyme and that colony does not appear the blue in color. With this feature, we can identify the transformed cells or not transformed cells. Suppose if that foreign DNA are insert outside the laxate sequence, they can produce the beta galactosidase enzyme and that colony appears blue in color. In another case, that insertion is not takes place, the recombinant DNA is not inserted into the plasmid, they are able to produce the beta galactosidase enzyme, then the colony appears blue in color. With this feature, we can identify the recombinants and non-recombinant cells. For screening clones which contain the recombinant DNA molecule, we are going to use a chromogenic substrate called XGAL. Chromogenic substrate called XGAL. Chromogenic substrate means they are get hydrolyzed and they are able to produce pigments, colorful pigments. That is why they are called as chromogenic pigment. So XGALs are added to the agar plate. So this is a plasmid vector. Please assume these plasmid vectors are introduced into a host cell, that is E. coli. Then these XGALs are added to that agar plate. If that E. coli produces, that host cell produces that beta galactosidase enzyme and hydrolyzed this XGAL, and able to produce 5-bromo 4-chloro indoxide. So that beta galactosidase enzyme can hydrolyze the XGAL and it produce 5-bromo 4-chloro indoxide, and they are able to produce this 5-bromo 4-chloro indoxides are able to produce insoluble blue pigment. Blue pigment. So that colony looks blue in color. So we are able to identify that colonies are formed by non-recombinant cells. If the bacteria, that E. coli, produces the beta galactosidase enzyme, that enzyme which can hydrolyze the XGAL, then that XGAL produces 5-bromo 4-chloro indoxide. They are able to produce insoluble blue pigment in the colony. Then that colony looks blue in color. So with that blue color, we can identify those colonies are formed by non-recombinant cells. Suppose if that colony not produce the blue color, if it appears white in color, that means that colonies are produced by or formed by the recombinant cells. Then we are going to insert or added the XGAL with IPTG. What is called IPTG? IPTG means isopropyl beta D1 thiogalactose pyrinoside. IPTG means isopropyl beta D1 thiogalactose pyrinoside. So this is added along with the XGAL to the culture for inducing the beta galactosidase enzyme. That is this enzyme, this isopropyl beta D1 thiogalactose pyrinoside can induce the operon to produce the beta galactosidase enzyme to hydrolyze the XGAL. So this substance or act as inducer. It can induce that operon to produce the beta galactosidase enzyme. So that's all about insertional inactivation method. Then I am going to tell about the next method, antibiotic resistant marker. Let me teach you how can we use this antibiotic resistant marker in the selection of transform and non-transform cells. So this is a plasmid vector which contains antibiotic resistant gene or the selectable marker. So this plasmid vector is transferred into a host cell. So the plasmid vectors are introduced into a suitable host cell. So this is the bacterial genome and this is the plasmid vector which contains antibiotic resistant gene. Then this bacteria are allowed to grow in an environment which contains the antibiotic ampicillin. So this bacteria are able to survive in this environment because it contains the antibiotic resistant gene. Then the antibiotic resistant genes are able to produce a protein which is resistant to the antibiotic ampicillin. So with this feature we can identify the recombinants and non-recombinants. Suppose if the bacteria do not receive the plasmid vector, the bacteria are not able to live in that environment because they do not receive the plasmid vector. So with this feature we can identify the recombinants and non-recombinant cells. Example for the antibiotic resistant genes are ampicillin resistant gene, then tetracycline or kanamycin antibiotic resistant gene, and chlorophenicol antibiotic resistant gene. So we can make use of the antibiotic resistant gene in the selection of recombinants and non-recombinant cells. Then I am going to tell about the third method, replica plating technique. Let me tell about the replica plating technique. Actually, this technique was devised by two scientists. They are Joshua Lederberg and Esther Lederberg in the year 1952. Replica means making exact copy. So in this technique we are going to make the exact copy of the master plate or the culture plate with the help of the sterile velvet cloth, and this structure is called as filter plate. So after taking the imprints of all bacterial colonies in the filter plate, so here you can find the bacterial colonies are present on the same relative position where they are present on the master plate or the culture plate. So this filter plate pressed against the culture plate. So this is the first culture plate which lacks ampicillin. Then this filter plate pressed against the second culture plate which contains ampicillin antibiotic. Then these two culture plates are allowed to incubate. After the incubation we can compare these two culture plates. In the first culture plate we are able to find all the bacterial colonies, while in the case of the second cultural plate we are not able to find all the bacterial colonies. Some of the colonies are removed by or destroyed by the antibiotic ampicillin. So we can compare these two culture plates and we can identify the transform cells or the recombinant cells. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.